All right, so the third objective is to compare the movement of molecules across the plasma membrane by carriers and through channels. And I keep reiterating this idea that while we speak about passive movement or passive transport, there are two different flavors of passive transport. So passive means in and of itself that it's moving spontaneously down the concentration gradient. One flavor of passive transport is simple diffusion, where it doesn't need any carrier or any help to get across the membrane. It can freely move across the membrane. And so that's what we spent objective 1 looking at, was basically what are some features of the membrane, what are some features of the particle that would allow for that simple diffusion. Now we're going to move on to look at also passive transport, but the second flavor of passive transport is where we now need carriers and channels to allow for that movement. The first example that we talked about on Wednesday was transporters, right? So for example, the GLUT4 transporter was what we ended off looking at, and that was a type of facilitated diffusion. And then the other two types would be carriers and channels. Okay, so we did speak about the facilitated diffusion, excuse me. So we're going to skip right over this. And then we're going to start looking at channels. So a channel differs from a transporter in that a transporter is more actively involved in that transport process, right? So it's going to bind glucose, for example. So the GLUT4 transporter will bind glucose on one end. It will have a configurational change, right? It's going to sort of shape shift, and then it's going to kick the glucose out on the other side of the cell. Channels, on the other hand, they're not actively involved. They're kind of just passageways. So they're just standing there passively, but they do allow for a channel, right? Just a pathway of movement for items or molecules to get through the cell membrane. So channels are going to be transmembrane proteins as well, right? So they're going to be always spanning both sides of the phospholipid bilayer, both leaflets, right? So it's going to go all the way through the membrane, and it would have to, in order for the job it has to do. It's going to function like a passageway or a pore, if you will. It's not going to have any enzymatic activity or any configurational change. It's just going to sit there as a pathway to allow that molecule to pass through. It's also going to be substance specific. So some channels will be specific to water, some will be specific to ions. And so while it's not going to be engaging with the molecule as such, it's going to be specific to one type of molecule or the other, right? So here's what the close-up of that channel would look like. It's going to have these polypeptide subunits. Remember, it's a protein, so it's going to have these different subunits which are coming together to make this opening in the membrane, and then certain items can just go right through. Now, again, I keep pointing out here, they're still going to be moving according to their concentration gradient, but they're just going to need this pathway as help to do so. Types of channels we have are aquaporins, and aquaporins help to move water. As the name would imply, these are really common in the kidneys. This is how we reabsorb water in the kidneys. So as water is filtered out, instead of us wasting or losing that water in urine, a lot of that is reabsorbed or reclaimed, right? We talked about reabsorption earlier on in the semester, and the way that that happens is by aquaporin channels. Other types of channels are ion channels, so channels specific to certain ions, and these can be leak channels. In other words, they have no condition. They just freely move ions according to that concentration gradient. Here, again, they're going to be specific. So specific to sodium, specific to potassium, calcium, chloride, etc. The other subtype of ion channels are gated channels, and gated channels need some condition to be met. All right. And when we move on to chapter 7, we'll talk about some of the subtypes of gated channels. So depending on what condition needs to be met, it could be a voltage gated channel where the cell has to get to a certain membrane potential, and then that channel would open up. It can be a ligand gated channel. This is where something has to bind, right? Some type of ligand has to bind, and that would activate that channel. So gated channels, there are a whole slew of subtypes, but they have a certain condition or requirement which act like a gating mechanism to open that channel. And then the third subtype are bidirectional channels, and then those, again, will just allow, as the name implies, bidirectional movement of ions. But it's still going to be specific to one or the other type of ion. Now, some of the factors that would affect the rate of transport required by ions or ion channels are basically similar to what we said for facilitated diffusion. So because we have a limiting factor, because we have a number of structures that can get maxed out, the weight of each channel transport will affect the overall weight of ion channel movement. The number of channels present in the membrane will also affect the rate of transport, and then the state of the channel. So let's say we had a bunch of ligand-gated channels, and they were moving at a pretty fast rate, right? They had a pretty fast rate of individual movement. But let's say they were closed. So that is certainly going to decrease the overall transport of the ion, even if you have a lot of the channels in the membrane, and even if they have a fast rate. So the state of the channel, whether it's opened or closed, will certainly affect the overall rate of transport through ion channels. Okay, let's look at these other checkpoint questions here. I'm going to pause for just a second, and I'm going to give you, let's do 3 minutes. We'll do 3 minutes. Write your response down, and then we'll share some of those and discuss.